Hello everybody, so my name is Rachel. I'm from Hannes's group here at MIT with Regina and Tommy, and today we'll talk about predicting targets. In particular, let's start with a motivating example. So this is a very classic image of a funnel that's used to motivate a lot of machine learning for drug discovery. It's often cited that this process is very long, arduous, costly. Of these costs and timelines, about 40% is spent in preclinical, so that's everything from the basic biology up to animals. And one of the main goals of these AI models for drug discovery is if we can condense this funnel, so if we can perhaps pick the right molecule at the very start or pick a few right molecules, then perhaps we can shorten the time and cost and drug development will be great. But there's a slight issue. So historically, finding the proper target for these molecules is also super hard. So even if we are able to condense the funnel through machine learning, there's the problem of finding the correct problem to target. And we still do have to try a lot of different targets in the clinic. Perhaps you only learn after the fact whether it's good or not before we find a real drug. So a complementary view in this work is if we can better select which funnel to work on at the very beginning, then this complements all of the machine learning for molecules that's going on to make drug discovery overall faster and cheaper. Okay, so the title of this work is as shown. So in the following slides, I'll first provide a brief intro to what are perturbations and targets, and as well as like what is causality, because this reading group is typically more, I guess, molecule focused. All right, so first, what are perturbations and why are they appearing everywhere in these bio-ML papers? As you can see, in the past 3-ish years, people are predicting perturbation effects. Perturbation-related things are quite common as a task for these foundation models or general frameworks. So at the core, perturbations are very simple. You have a set of cells, you do something to them, so you can add a drug, you can use CRISPR to knock down or knock out genes, and something happens to the cells. So that word broadly refers to an intervention in the context of molecular biology. And after we do something to the cells, it's quite common to read them out in one of two ways. The first is imaging, which we don't discuss here, and the second is known as omics. So that means inside each cell, there are many biomolecules. Can we somehow quantify them to describe the state of a cell? So what is going on? In particular, this work focuses on quantifying the RNA inside of a cell because it's easy to measure and it's possible to enumerate all the potential genes. So it's, I guess, unbiased in this sense. And why are they becoming popular? Well, within the past, now it's been 3 years, it's become possible to do these perturbation experiments at very large scale. So since the genome-wide perturb-seq paper, people have been doing very large perturb-seq, i.e. Using CRISPR to knock down genes experiments. And for a sense of scale, we can intervene upon as well as measure about 20,000 genes and up to hundreds of thousands, millions of cells, or even more. So this generates very large data sets that are quite nice for machine learning problems.
In these data sets, do we then always have one perturbation per cell, or is it that multiple perturbations are, or multiple genes?
So yeah, it's more common to find one perturbation per cell. It's experimentally possible to do more, but because the number of sets of perturbations scales combinatorially, people haven't quite done sets of perturbations at scale.
Okay.
Yeah. All right, so to summarize, genetic perturbation screens, i.e. perturb-seq, lets us poke cells and measure them. We can do this at very high resolution and scale. Okay, so what do perturbations tell us about biology? So we have our cells, we measure the cells, and we get a data set where every row is an individual cell and every column is a gene or, say, a protein. This can happen at the resolution of single cells or more traditionally at the resolution of groups of cells. And one thing you can do with this data set is you can compute the correlation between all of the genes and themselves. And you'll observe things like X and Y are related. So perhaps X is the protein target of a known drug, Y is a disease marker, and if we use a drug for X, then hopefully perhaps we want to impact Y. And just measuring things will tell us that these two genes are related, but it doesn't give us the causal direction. So by actually running the experiment of perturbing Y, we can look at whether X and Y are still correlated. And if after the perturbation they still are, then perhaps the relationship is causal. And by doing this at scale, we get a sense of many, many causal relationships between all the potential genes to perturb and all the potential readouts. All right, so the summary here is by doing all these experiments at very large scales, we are able to get causal insights about how biological systems function, also at scale. Okay, so the final part of the intro is, what can we do with this data? So there's two primary directions that machine learning works take. The first is, can we infer the effects of perturbations that we have not done or perturbations in novel contexts? So here the goal is simulation. And you could use this to say, predict how a system might respond under new conditions or predict how an unseen system would change. And the motivation here is because it's expensive to do these experiments in every single setting to perturb cells, cost scale as like the number of rows that you have. So if we can simulate this, then perhaps things can be cheaper. Now, this is a hard problem. There are many works, but overall, it's quite unsolved. So an alternative direction is we have some starting point, say healthy cells, and we have an ending point, say a set of disease cells. If we can figure out what are the root causes that take us from the healthy population to the disease population, then perhaps we can identify useful targets for reversing this change. So the goal here is we want to design experiments more efficiently because and we want to understand like what are the actual differences between the systems because many things may look different on the surface. And we want to do this through machine learning because, as we mentioned earlier, there's a combinatorial space of all of the perturbations that we could try, and it could be intractable to try them all. So this work focuses on the second direction.
So the second thing, we basically have a bunch of observations and then either other observations or like a desired, or I can say, I now want to instead have this distribution of observations. What do I have to, or what should I poke to obtain this distribution of observations?
Yeah, exactly. So obviously you would confirm it through experiment, but if you had to try everything, it could be slow.
Okay.
Yeah.
Now, how would this look like in practice for someone doing, I want to do some drug discovery now. How do I decide what distribution I want?
Yeah, so in drug discovery, a very common application is you have some drug candidate and it does something to the cell, but to be more confident that this is going to translate to actual humans, you perhaps want to find the target, right? So the thing that people currently do is they try knocking out many genes and they look at what is most similar to the drug's effect. So yeah.
Okay, so you would have your distribution before having the drug, your distribution after having the drug, and then you find out these are, my system now predicts that these are the causal variables that, or the interventions, the targets that could cause this new distribution, and then you go ahead and test them for whether they're actually the target of your drug.
Yeah.
All right. And so what does the guy in the chat ask? Can you please also comment on the reproducibility of these experiments? For example, between individuals.
Yeah, sure. So, I mean, I guess in general, if you measure the same cell, if you have the same intervention, you do it twice in the same cell line, the same person does it, there's actually pretty low correlation between the readouts that you're likely to get. So a lot of these experiments are interpreted at coarser level, say like this gene impacts translation, or I guess that gene impacts a certain pathway. So I guess another way of describing this is you have some very coarse target, you know that translation is messed up in these cells, you want to figure out what are the potential explanations for why, I guess.
Okay.
So I guess the idea is, for this data, it's perhaps a bit better to operate at coarser things than like what is the real value change in a gene, which, as the person mentioned, is not quite comparable often.
What are these coarser values?
I mean,
how do you obtain the coarser values from the finer values?
A lot of it's based on existing literature. So one paper writes, these genes are involved in translation. We observed it through like some other data modality, and then this becomes a gene set that people use in the future to sort of cluster genes.
Okay, and then whenever like the interpretable distribution, so to say, is I get all my gene measurements, and then I like take the average of the gene set that is responsible for translation, and then that is my coarse-grained translation.
I mean, so this is more relevant in the first type of application, but you would say what genes are changing. I look at these genes and I say, okay, these genes all look related to translation. It's not really real valued, it's more at the discrete level.
Yeah, and you're saying it's not really relevant here because here I can, or less relevant because here I can just take the discrete, the real valued stuff, the other fine-grained real valued stuff, and then compute my interventions on the fine-grained level and do my confirmation knockout experiments on the fine-grained level.
So yeah, I guess in this paper for the evaluation, it's on predicting the target. So we can only evaluate in cases where we know the target. And if we know the target, then the label is very clean because it's part of the experimental design. And the variability comes in for actually, the first class of methods because they evaluate on the level of these readouts rather than the interventions.
Okay.
So it's quite related to the application, but not quite.
Yeah. And the interventions are clean, so what we're evaluating on is also clean.
Yeah, because you know what you put into the cell, because you did it. Okay, so going back to, I guess, the idea of searching for targets, the very classic approach is, as we mentioned, you try many different perturbations. And you could be lucky and you could find the actual perturbation that induces the change that you want. You might also be unlucky and you tried many things, but you never get close. So this can be costly and this can be inefficient. The alternative approach that we take in this paper is just if we think about it fundamentally, why are these two distributions different? Well, it has to do with like the causal mechanisms. So it could be changes to the gene regulatory graph that induce these differences. So these changes that we do not observe result in the differences in the data that we do observe. Now, based on this intuition, we have these different, I guess, graphs which are latent. They caused the differences in datasets, which we see, and our goal is to find the targets. And based on this idea of the graphs, if we can somehow invert the datasets back into the graphs, so if we can predict the causal mechanisms or the gene regulatory networks from each of these datasets, then it should be relatively clear to infer what the actual targets are. And this is because, like, at the level of these causal mechanisms, we do not, like, the things that are impacted downstream do not change the fundamental causes, whereas the things that are changing do. All right, so on a high level, we have two modules to our method. The first is we predict causal graphs from datasets. So this first module is called a causal graph predictor. Now, the way it's implemented is quite, I guess, intuitive. So if we take the raw data and we put it into a neural network, it can be very large because there could be millions of cells. Each cell has 20,000 genes. It's quite difficult to scale to this size. Instead, we represent the data in terms of summary statistics, which is also how biologists interpret this. So first, we have global statistics, such as pairwise correlation, or this in the biology language would be co-expression. And this gives a coarse estimate of what relationships might or might not exist. And the second feature is we look at these local estimates. So we take very classic causal discovery algorithms, which produce graphs. We run them on small subsets of nodes or genes that are likely to have relationships, and this yields edges which have directionality. So these two summary statistics are input into a neural network, and this neural network, based on attention, outputs a graph. Now, this is trained in a supervised fashion where we assume that we have access to ground truth graphs. And this is via data simulation, as I'll introduce soon. Okay, so first we evaluate this graph prediction module on this data set called causal bench. So basically the goal is to predict relationships between genes where the ground truth is based on knowledge graphs, i.e. data mined relationships. Oh, sorry. The question is, are we doing single cell or bulk? This is single cell, so we can compute all these pairwise relationships.
What is bulk?
Bulk is you only have one measurement per population, so it would be harder to get correlation. You would have to have multiple subpopulations, yeah. But then, so this graph prediction module, we evaluate it here, and it runs very quickly. It also has very good precision. So the relationships that it predicts are actually present in the data.
Can I ask one question, please?
Yes. Hi.
Hello. Can you please go to the previous slide, please?
Yeah.
So just to make sure I follow like the algorithm, like D is the initial like gene by cells that you mentioned, like in the beginning, and sorry, D, and then G is the perturbed one. Like I'm trying to understand the mapping here.
Oh, so we don't have the distinction between perturbed or not yet. So D could be it could be control, it could be perturbed. The same thing will happen to both. So G would be the gene regulatory network.
Okay, thank you.
Yeah. Right, so going back to this slide, each of these like perturbed and control will have its own network, and these should be different. All right. Okay, so we evaluate the graph predictor. It seems to do okay. It seems to run quickly. So now we can use this graph predictor and run it over each of the data sets. So once over the control, once over the perturbed. And this gives us a set of features related to the causal graph or the gene regulatory network. And we also compute additional summary statistics like mean and variance. And this is inspired by the bioinformatic approach of, say, when you look at differential expression, you look at what things change and you scale that based on the variance of the gene. Okay, so we have two sets of features. They go into an additional model, which we call a differential network, whose goal is to predict targets. And this differential network is implemented using a similar attention architecture as the causal graph learner. It's a bit deeper, but basically we either concatenate or take the difference between these sets of features. We run attention over all the rows and columns for a number of layers, and this is aggregated to predict targets. Now you might observe that the training of these models requires the ground truth. So we obtain fake ground truth through data simulation. This is because it's quite hard to get actual graphs and that correspond to actual data. So we simulate lots and lots of graphs. We simulate lots and lots of targets. This can be via, say, RNA simulators, or it can be more classical approaches where we have, say, we simulate linear relationships, we simulate nonlinear, Gaussian, non-Gaussian, various graph topologies, and so on. So these fake data go into training both the graph predictor and the differential network.
I think you have like 7,000 synthetic graphs, right?
Yes, so there's 8,000, some number of thousands of synthetic graphs that are all different.
Why only 7,000?
So when we sample different, I guess, data points. We sample different sets of nodes, so there's stochasticity there. And it's about as much as will fit nicely and load quickly on one server.
Okay, so these graphs are quite big, and when training you, there's some randomness in how you sample them anyways.
Yeah.
And you sample like subgraphs of them as well, or something like that.
Yeah, so whenever I sample, I vary the budget of how many points I want to sample, of how many little estimates. So even though we're repeating the same data set, we're sort of seeing different input featureizations, because if you compute the correlation of like different subsets, there's slight variability.
And in your experiments, do you sometimes not fine-tune and only use synthetic data?
Yes, so yeah, right.
Unseen experiments.
Yeah, so if you're able to fine-tune on real data, like perturb-seq data, then you can do so. In this case, we freeze the graph predictor and we only train the differential network because we don't have the ground truth graph. So here, say if you did perturb-seq in one cell line, you can fine-tune on that and apply it to another cell line. Empirically, I found that fine-tuning on perturb-seq is not good for predicting drug targets because perturb-seq is much more specific. So you have the option of doing this or not doing this. Yeah. And also previously for the data simulation, we can do things inspired by, I guess, biology. So you can log fold change can be measured in, I guess, log space or original space. So for the perturbations, you can add or multiply by some factor to obtain what the perturbation should be. Okay, so the experimental setup is we have two settings. One is perturb-seq, so we're manipulating the genes. And then the other is known as cyplex. So this means we apply drugs to cells. Now, given the control plus the perturb, we want to predict the target, which we know as ground truth. Now, to do the data splits, because many perturbations might have similar effects, we first compute the log fold change of each perturbation. So this is pseudo-bulked over all cells of the same perturbation. We cluster them, and then we split based on the clusters so that the train and test look relatively different. And for citations, these come from Replogo et al. And Nadig et al. These are, I guess, data sets with 1,000-ish to 10,000-ish perturbations over 100,000 to around 1 billion cells. Okay, so for the perturb-seq experiments, we want to emulate having to find the actual target. So the thing that we measure is how many perturbations do we have to test before we find the true one. And we take the top P% of the perturbations and see whether the true target is actually within the top P%.
So you basically have a rank of all perturbations, and you compute the average that you need to get the.
So from left to right on the axis, we have like take the top 0% versus take 100%.
Yeah.
And we take the average overall perturbations of did we find it or not.
Yeah.
Yeah.
Well, how can something be? So if I just randomly put my thing, like if I just randomly shuffle it, then I would have the diagonal, no?
Yes.
So how does, for example, the blue line and have G2 achieve being worse than that? Like consistently?
So that one is, what is it? It might be GPT or, yeah, I think it's GPT. If it's consistently worse, then, I mean, I guess the AUC would be less than 0.5, right?
So just invert your ranking.
Yeah, if you knew in advance.
No.
But for the K562, it does okay, so. Yeah.
Okay. Well then, so the top 2 lines are, or there are 3 lines at the top, all right? And 2 are from you,
yeah.
And the third one is which?
Okay, so the top 2 lines are our method. The top line is if you fine-tune on perturb-seq. The bottom top line is if you only take simulated data. And the third line is the thing that people currently use. So differential expression, i.e. you have your 2 distributions and you test statistically whether there's a difference in this gene. And if there's a difference, based on, I guess, the p-value, we rank the genes and see whether that's the target or not.
And why is that slow to run?
It's very fast to run.
Okay.
It's what the biologists use because it's very interpretable and it's quite fast.
It seems to work very well.
Yes, and yeah.
So then the scenario where we're able to train on like half the data of the graph, is that actually realistic?
So I believe these are training on different cell lines. So I think it could be realistic that you have perturb-seq data from a different cell line because we already do.
From a different cell line, but
yeah.
Here you always train with the same cell line, no?
Oh no, so there's one setting where we have train test splits, within cell lines.
Oh, okay.
And we have all the trains, all the tests. And the second setting is we hold out cell lines and we train on the trains of the others.
So here you hold out cell lines.
I think this is that plot. I need to check.
Okay.
Alright.
So do the plots look similar then? Or
I think they look quite okay. The table looks quite similar.
Okay.
So.
But then like training on different cell lines, I thought you only have like 5 cell lines or so.
So I have 5 cell lines, but these cell lines are public. So when you have a new one, you already have these.
But so I'm supposed to believe now that from training on 4 cell lines, we can generalize well to a 5th cell line. Is that what's going on?
That is the.
Oops. Very good. Monchichi scared the visitors off.
Yes, the hope is by training on these existing data sets, you can generalize to new cell lines.
But like from training on 4, I'm supposed to generalize to the 5th one.
Yes,
This
so
sounds like not a lot of training data for.
I mean, so each of the 4 has like 1,000-ish perturbations. And also in this model, it's based on these sort of statistical features. So I'm not actually saying this is like the gene. It's based on did the distribution of this gene change. So because it's more meta information, like I think it's more realistic that you could generalize.
Okay.
Because like different genes are up or down in different cell lines. Yeah. But like the characteristic of did the distribution shift could be more transferable.
Okay, makes sense.
Yeah. All right, so briefly, the baselines are PD Grapher, which is quite dark, is it's a machine learning model. It's a graph neural network that does this task of predicting targets. Gears and GPT, these are baselines that predict the effects, and we look at which predicted effect is closest to the actual thing that we're trying to find. Wow. So overall, it's quite hard for.
Well, then GPT must be very slow to run, right? Because you need to test every single possible perturb or every single target and then see what effect it has.
So yeah, Gears is slow to run. GPT is an LLM embedding model, so you just train, say, a logistic regression. So GPT is very nice and fast. Gears is kind of slow because you have to fine tune.
Okay, but in both of them, I need to test every single possible gene for whether it achieves the observed effect.
Yeah, so, but if you're just running GPT, presumably because it's just the logistic regression, it could be fast.
Yeah. Okay.
Yeah. All right, so this is perturb-seq. And then for the drug targets, we looked at a smaller set. We looked at genes in which the drug target is actually differentially expressed within the top 1,000. So this is, I guess, a pretty lenient threshold. The baselines are differential expression and the PD Grapher, so the GNN predicting targets baseline. And our model is able to predict the ground truth target in 4 of the 6 cases. But because I guess there's noisier data for drugs and the real targets are less clear, I guess these evaluations are smaller. So we need to try it on more. Okay. Yes.
That's it?
Yes. So now I guess we have discussion of applications. So in the evaluations, we looked at cases in which it's very clear what the target is because it's part of the experimental design. But we want to actually use this in cases where we're not sure what the true target is. So there's 3 primary applications. The first and second are, as we discussed, we have healthy and disease cells. We want to know what drives the disease. And the hope is by understanding the mechanism of the disease, we can better treat it. The second is deconvolving the target of a drug. So when you apply a drug to a cell, drugs are known to be very messy, i.e. they can impact thousands of genes. And we really want to figure out what specific target or targets it affects. And then the final application is for cell engineering. So perhaps we want to generate heart cells to be used in therapies. But we start with stem cells. So the start and end would be stem cells versus differentiated cells. And we want to find which of the several hundred transcription factors or what set we have to apply to achieve our target state.
Or maybe the other way around. Like I have some cell and I want to turn it into a stem cell.
Well, there are 4 transcription factors that seem to do a good job. So I guess people are decently happy with that direction at the present moment. Yeah. Okay, so that is, I guess, what we have to discuss. These are the 2 papers. If you would like to learn more, the first is this paper, and the second is the causal graph prediction model. And this is joint work with these lovely people from our group. So yeah, thanks for listening, I guess.
The second one is essentially where the architecture for the causal graph prediction comes.
Yes, this has the annoying transformer that we had to code.
Okay.
And the first is a Q application.
So I guess we have some questions from my side at least.
Yes.
Yeah, in the beginning, you cite some papers or in the background, no, not in slides, about how these models that predicts effects instead of you now predicting targets, they don't really work for identifying targets. Okay, so these are the go-to models for that.
So yeah, this one, the two on the left are go-to models. I mean, there's multiple single-cell foundation models, but they are the go-to for predicting effects of perturbations.
Are
Simulating.
Are they good at predicting effects at least? If maybe the effects aren't good enough for going ahead and making the reverse inference of being the target that caused the effect, but are they good enough for at least some uses?
They're good at identifying what genes tend to change or not, but they are bad at distinguishing, like if you compare two very similar cases, in one case it changes, in the other case it doesn't, they're bad at distinguishing this fine-grained stuff.
Okay.
So in the inferring effects task, there have been some papers that say predicting the mean of like whether this gene tends to change across all perturbations is a super strong baseline. So I guess there's a bit more precision needed for these models to be super useful, at least in my opinion. Not to say that they're like bad, but I think there's some ways to go. Yeah.
Have you seen this new paper from Bo Wang?
Which he has many new papers.
I just saw it this morning on the Twitter's paper from Bo Wang. Maybe let's have a question.
There's an LLM paper he wrote this morning, but
yeah.
There are, yes.
Maybe let's have some questions from the audience. So Nicholas, unmute if you want to go ahead.
Sure, yeah. I must admit, I don't know if my question will make much sense, but I was wondering, like, because you said when you fine-tune the model, like, based on actual data, you might not know the graph. If we have a target in mind, can we try, like, to just use practically like the back propagation or like auto differentiation to get an idea of what the graph might look like, the importance might look like? So in other words, can we use the same model to understand something about potentially the structure of the graph or not really?
Yeah, you can. So we don't train the graph predictor for real data, but we do. We do still have the graph prediction head, so you could still take the graph that comes out. And it's sort of like the differential network is sort of tied to the graph predictor because we do not change it. So hopefully it still does make some sense. So yes, I guess you could look at it.
Is it something that you have looked, like if it performs reasonably well, if it's explainable or not yet, perhaps?
Oh yeah, so for the graph predictor, that the experiments would be in the graph prediction, so the previous work, but we have looked at it in the context of recovering what's known in knowledge graphs, and it does seem to be reasonable. It has high precision, it has lower recall, so I guess that's sort of like the slight caveat. Yeah.
Okay, thank you.
Do effect prediction models predict on the aggregate, on the gene set level, or on the gene level?
They are typically trained on the gene level.
Okay. And used on the gene set level?
And also typically used on the, I guess, gene level. At least in evaluations. I don't know what's actually used in pharma.
Like how, right, this sort of model is kind of more if you had the perfect model, then it would be more capable than just a target prediction model.
Yeah, if you had a perfect model, you would have what Stanford likes to call the virtual cell, right? And also, like, sometimes these models are evaluated on like what is the neighborhood of this gene. Does it make sense? So there's like evaluations that consider a variety of aspects.
And then is this task actually, or would you say the majority of how pharma currently tries to experimentally do like things related to targets, to mechanistic understanding of what the target is for this and that, or trying to identify new targets using things where you need to find out what the target is, or things where you want to predict ahead what, like where the effect prediction model would be necessary. So basically the question is, if we had your model perfect and we had the single cell foundation model perfect, then how much would it actually be valuable to have the single cell, the perfect effect prediction model instead of the perfect Euro model?
So I guess it's slightly out of the scope of this work, but in general, like if you had a perfect model that could predict the effects of perturbations, you could presumably identify new targets, right? Currently in pharma, to the best of my knowledge, pharma the majority of pharma does not focus on identifying new targets. It's more based on literature review of what have people looked at and what are like hints that we could pursue further. But if you had the perfect model, you could start going in yourself and finding the targets. Okay. And then the second, so my approach is more applicable to, say, phenotypic screening. So you have a coarse readout of did the cancer cell die or not? Did I, based on this image, achieve some desired effect? And here, deconvolution is quite, I guess, people want to deconvolve the target even when they have the candidate, because if you can do so, you have more confidence that the drug will work. So my model will be useful there.
Can't I also do target discovery with your stuff when I once have a cell population that's not cancer and once that is cancer, and now I go ahead and try to find out what are the targets or the interventions that make the cell cancer, and now I have identified the targets that I want to deal with in my drug discovery to treat the cancer.
Yes. So I guess, yes. And so there's two, I guess, light things. Yes, you could do that. My model, I guess, would, so if you have healthy and disease, then my model would be useful for, say, like, what are the things that cause the cancer? And you would be inferring that, like, if I target them, then like, presumably the same things that cause cancer may reverse it. But this is not guaranteed.
Okay, but like, isn't it always the case that if there's a disease, then like it is some gene of some protein is affected, and that leads to some causal chain that changes the overall cell state, and yeah, then if I find out what is the thing that has been affected, like what caused the change of the cell state, then I've discovered the target that I need to treat the disease.
So yes, this is a huge motivation for, I guess, this work.
Okay.
But I do want to add a caveat that, like, sometimes a different gene will sustain the cancer versus initiate it. So, like, there are nuances, I guess. Okay. Yeah. But yes, we wish we could do that.
But then, yeah, okay, fine. Ravi, or maybe not. Did I have another question?
Yeah, I had a question. Can I ask my question? Or?
Yeah, go ahead.
Yeah, thanks. Thanks for the talk, Rachel. I really appreciate it. I just wanted to ask about the neural network that was in your slide. So, and forgive me if this was already mentioned. What kind of neural network did you use in your paper? I just want to
Yeah,
understand.
I can show you. So there's more details in the paper that describes it, but I'm not sure if it's visible here. So the inputs are these two sets of core statistical features, right? And they are each embedded, similar to, say, word embeddings for the edge types, and just a linear projection for the statistic. And after they are embedded, there's a series of 2D attentions on each of the features. So this would, on the left, we would have like number of edges times number of estimates. On the right, we would have number of nodes times number of nodes. This can be, both can be quite large. So we attend, I guess, along each dimension, along each axis separately, and we have these sort of message passing operations that pass information between the two representations. And at the very end, we take the m by n for the graph prediction.
Okay.
Now I
I understand
remember that you also have this FCS algorithm also that you run in the beginning to obtain estimates of something. How expensive is that? And do you think your your causal graph is not actually anything more than a stitching together of these local estimates?
So the FCI algorithm produces the local estimates, or in this case, you could also use things like GIES to consider interventions. These are quite cheap, I would say. The whole thing runs in like about a couple seconds on larger graphs. Is it just stitching it together? So what I noticed in the ablations is that if you have fewer estimates, or if the estimates are noisier, then the global statistics are more valuable. When you take them away, the performance drops more. If the estimates are higher quality, so GIES with interventions produces pretty good estimates, then the global statistics matter less, and it's more about the stitching. So it's the model sort of learns which to trust more.
Okay. Then last thing, you predict all the targets independently of each other.
Yes.
Is that because you always only have one, or in your data you always only have one perturbation per? No, well, one target. No, I don't know. Why don't you do it autoregressively or something?
You could. I just didn't. Yeah.
Okay, you just did.
But
also
you
didn't.
could. Yeah.
What's your confidence that it'll help?
It probably will if you have like a main target versus weaker off targets for a drug. It, I guess, makes sense to do if you already know one and you condition on that. I was lazy, I guess. So it's currently independent.
Then,
all right.
Friends of Sun, any last questions? This is an ICML paper, right?
Yes.
Okay, then.
And say hi next month.
Next month.
Oh.
We have someone's commenting, I think, a wave. It's not a raised hand as far as I know. So, any last words from you, Rachel, before we end it? I guess.
Wait.
All right, Denise, let's go.
Yeah, thank you. Thank you for the presentation. My question is, I guess, about the causal subgraphs. I just want to understand a little bit more the directionality that you impose. It is local, I believe, right? So you do not create like large causal subgraphs, but only in the vicinity of some gene. And do you have some way to first kind of filter out regions of candidate genes, or do you do this unbiased in an unbiased way?
I see, yeah. So I do a mix of both. So you could use, say, co-expression or correlation to identify clusters that you should be running the estimates on. You could also just sample unbiased randomly. So I guess in the original graph paper, I did half of each.
Mhm. And so I kind of have two questions. First, is you, I believe, using purely expression data, so your correlation graphs are based on expression, and this way do not really, I guess, capture like mechanistic interactions between genes, right? So it's more on the level of reactivity of a cell to perturbation of the gene, correct? So like you kind of rely on the fact that if in a given signaling pathway one gene becomes overexpressed and overactivated, this will result in downstream gene becoming regulated by expressivity.
Yeah, so the input is just co-expression. If you had, I guess, graph priors or mechanistic priors, you could incorporate that as an input to the model. You could either include it as sort of, I guess, an extra subgraph estimate or fine-tune the model to take priors. But yeah,
Yeah.
the current model only takes the co-expression.
And am I right to remember that gears actually do use directions based on some perhaps mechanistic interaction between proteins? Or is it not the case?
Yeah, so Gears has two graphs. The first is co-expression, the second is gene ontology. Um, the gene ontology, I guess it's hard to say whether it's super helpful or if you should include more or less information, because in Go, there's lots of relationships, like this regulates, this does not regulate. But for Gears, it's all collapsed into adjacency relationships. So I guess given sufficient data, perhaps you could learn the semantics back. But as implemented, I would say it's a bit minimalistic and could be more interesting.
I see. So
Could
I
include
guess my question, like the actual question that I'm interested is if your the graphs that turn out to be, I suppose, I'm not even sure if they are the drivers of good predictions for a model, but if they are, then maybe you can observe that their structure corresponds to some particular type of relations from gears. So like you kind of got some interaction between proteins, and maybe it corresponds to some particular type of mechanistic interaction. Like maybe you observe that this graph maybe can be explained by some kind of like phosphorylation pathway getting overactivated and so on. Maybe this can be tested by taking the input causal subgraph that leads to good activation and then comparing it to some activated region in the gears network or something like that. Like mechanistic analysis of what actually these causal subgraphs are doing.
Yeah, I think that would be pretty interesting. Yeah, and for the graph evaluation, the data set did take stuff from, I think, like Gears string to construct the sort of ground truth. So that captures a bit of it, but I think more fine-grained analysis would be interesting. Yeah.
Yeah, okay, thank you.
So Theophanes Karaletsos says, I think this when prodding him on the Twitters, I think this may well work for some targets, but I also have not spent the time to read the work. Fundamentally, the closer we get to trying to model a mechanism of action, the more likely we will generalize usefully. Modeling forward effects via a foundation model, FM is foundation model, not flow matching, just mimics screening and this is why it is popular. What I sense you're asking here is if we could skip screening and directly go to targeted probing of interventions. My questions would likely end up being anchored in practicality of aligned lab strategies for validation, if I understand correctly. What? Anchored in practicality of aligned lab strategies.
I guess
Get what he means?
maybe is it easier to sort of simulate lots for your experimental design, or is it easier to use a predictor?
Okay.
I think.
Well, and Anshul just says, I really like this idea. Need to read the paper in detail, but I'm definitely far more optimistic about this kind of strategy.
I mean, they're different. They're complementary, right? You can have both. Ideally, we have both.
Well, Anshul is known for not being a big fan of the, let's say, effect prediction and just saying that they're...
If it could work, he'd be happy too.
Yeah,
if it
yeah.
could work.
I don't want to misportray Anshul here, but that's my medium confidence impression. All right, then, friends of the sun, let's call it a day here and see you around.